I would like to talk about the forgotten thermodynamics and kinetics, and I did a review of these lot of publications, about 24 years of work, and at the end I found a new, or I would like to present a new model for the function of ABCB1. Now, we are all under the spell of these wonderful structures, and we kind of forgot about the physical chemistry and the kinetics. And here we saw these nice structures and constructed a model. And it's actually Kenny Linton and Higgins who first proposed this model. They saw in their crystal structures, faint crystal structures, since at the beginning they saw these A-shape and V-shape transporters, and they said that's a switch. We have no ATP bound, we have an open conformation towards the cytosol, and we bind then the molecule goes in through the aqueous channel, and we have a switch and the molecule can leave to the outside. And these are the structures of P-glycoprotein and of SAB, and these structure kind of confirmed this model, or seemed to confirm this model. And my question was then, where is the membrane, and what is the role of the membrane here in the middle of these transmembrane helices? What does it do? And we know that the first publication of P-glycoprotein was by Giuliano and Ling, and he identified the transporter, and he found that the substrates were all amphipathic or amphiphilic. And there are more things to remember. We have a physiology. We know that the ATP concentration is pretty high in cells. We have homeostasis. We know that the Km value is pretty low, which suggests that we have practically always in the resting state 2 molecules of ATP bound. So this structure, which assumes open conformation with no ATP bound, is for me difficult to understand. Then we have a second point, the nature of the membrane. It determines the interactions of the amphiphilic molecules which penetrate into the membrane. And there is a lot of work by Michael Gottesman group's early work, who showed that the allocrites bind through the membrane to the transporter. They are the membrane is hydrophobic. It's anisotropic, which means it is structured. It's fluid, but it's still, there is a structure. It has a certain packing density of these lipids. And very importantly, it has a dielectric constant. So the dielectric constant of the membrane is very low. Then we have kinetic factors, and we will see whether these, we had in the very early PGP titrations were all biphasic, and with time the inhibitory phase disappeared, and we will now see. So, and we will see whether this is real or not. Now the membrane has leads to a strong accumulation of the substrates. We have here a simple model of the transporter. The molecule is bound, it is exported, and in parallel we have passive flux. We have substrates which are amphiphilic. I showed the molecule in the way I would sit at the air-water interface. Air is hydrophobic, like a lipid membrane, and water is, of course, hydrophilic. So you see these encircled groups, which are hydrogen bond acceptor groups. And we found certain patterns. The first one is colchicine. And colchicine, you see, has a pattern with 3 atoms in it. And we proposed quite a time ago that these groups are inducing P-glycoprotein. Then the second one is a cancer drug, which is very hydrophobic, very small. It penetrates very much into membranes. And the third one is rhodamine. And this is a related compound. And the difference in the C and D molecule is that we have 2 patterns in rhodamine, but we have only 1 small pattern in the analog. And the reason is that the analog has a NH2 group instead of a tertiary amino group. And these NH2 groups do not form patterns. So patterns are only formed by tertiary amino groups, tertiary or quaternary amino groups, which have a positive charge. Now, this is a very early slide, which just shows the distances between the patterns. So we have 2 types of patterns, a type 1 pattern with 2.5 angstrom and a type 4 pattern, the inducer pattern, with 4.6 angstroms. What do these patterns, what are they good for? So here is a transmembrane part of the P-glycoprotein. And you see very many hydrogen bond acceptor groups in these transmembrane helices. And surprisingly, this always intrigued me. They look very much to the outside, not so much to the small channel. And so the role of these groups here, do you see my cursor? Yes. The role of these groups is to attract the hydrogen bond acceptor groups of the substrates from the membrane. Now, the possible interactions are hydrophobic interactions, electrostatic interactions, and they work if the molecule is in the aqueous phase and binds to the membrane. Then within the membrane, we cannot attract a molecule by hydrophobic interactions because the most hydrophobic medium in a cell is the membrane itself. So you cannot take it out by hydrophobic interactions. However, you can extract it by weak dielectric interactions, like cation P interactions. That's the most efficient one. We measured 6.5 angstroms, 6.5 kilojoules per mole for this interaction. The hydrogen bonding interactions are 2.5 to 4 kilojoules per mole. So people who have read early literature about hydrogen bonding would think this is a little bit low because one said 10 kilojoules or more. These are the calculated values, but I found recent literature, not very much, but I found some references where they measured hydrogen bonds in real membranes in a protein, and they found the same range as we found for the interaction between the allocrite and the transporter. Then we have pi-pi stacking interactions. That's the phenyl group, which slide over each other. And we have, of course, van der Waals forces. The 2 red ones are the most important ones because p-cation interaction, a very highly charged molecule, will not penetrate the membrane. So this is more for theoretical interest than for practical interest. And then the next point is the kinetics. We measured ATP hydrolysis by the classical method in inside-out vesicles, but also in cells. And for this purpose, we used the NIH cells. We are very grateful to Michael Gottesman that he has given us these cells, and we measured the ATPase activity as a function of concentration. Now, if you have low concentrations, we have the activation. If we go to high concentrations, we have inhibition. And this shape was discussed a lot less in papers actually as in small talk, I would say. And the second part here was a bit discussed. So is it aggregation which leads to this decrease, or is it is it too little ATP available? Is it a disturbance of the membranes? And we tested all these individual facts by physical chemical techniques. We did for the disordering, we did deuterium NMR. For the aggregation, we did light scattering. And for the exhaustion of ATP, we did ATP regenerating assay. So we are very sure that the down regulation is real, and we could not explain the novel PGP inhibitors without having two molecules bound to the transporter. And actually, it's now also proven by crystallography that two molecules can bind. So this is a very important factor. And then is an interesting experiment. We said that the membrane shapes the interactions. Now, the diffusion into the membrane decreases when you have a high packing density, because you have to invest more energy, or the molecule has to invest more energy to penetrate between the lipids. On the other hand, the binding directly within the membrane, the binding of the allocrite to the transporter increases if you have a higher packing density. And this is because the binding, the weak electrostatic binding, is very much dependent on the dielectric constant of the membrane. And if it is very low, the affinity is high. If you have high affinity, the rate is low. So we have on the left side, we have a membrane. We have the transporter in the membrane, and we have a relatively low rate. Then we put the transporter in a mice cell, and we get a very strong increase, almost threefold increase in rate. And this can be explained by the dielectric constant change. You can quantitatively calculate this effect. And interestingly, Suresh Ambudkar has done quite a big analysis of ATP hydrolysis in a very recent one in vesicles and in cells, and he found a very similar threefold increase when he goes to mice cells. And this was particularly interesting because he did it for these novel inhibitors which bind as dimers in cells. When he put them in mice cells, he got activation. So he had a switch from inhibition in cells to activation in vesicles. Now, we come to our suggestion. So we summarize here first we have the binding step consists in two individual steps. First, we have hydrophobic interactions. So we have penetration of the molecules based on hydrophobic on the hydrophobic effect into the membrane. In the cytosolic leaflet, we have in addition electrostatic interactions, which are, however, not very pronounced because we have a lot of salt in the cell, magnesium, which shields the surface potential. And so this is the first step, hydrophobic step. The second step is these dipolar interactions in the very low dielectric environment. In the middle, it's about epsilon equals 2. In the interface, it's about 30 to 40. And in the aqueous phase, it's 80. So from here, from the aqueous phase to the middle of the membrane, we have a factor of 40 increase in binding or in attraction. And the blue part here reflects the polar groups, and the yellow part, the hydrophobic part of the molecule. And our suggestion is that these molecules are attracted to the surface of the protein. We have two molecules. I should say, we also did molecular dynamics simulations, and we found that the molecule closes. We have always these open structures because we have no ATP when we do a crystal structures. If we add the ATP, we have a closing of the structure. So our starting point is this resting molecule with two ATPs bound. The allocrites bind to the transporter. At some point, it probably gives a distortion a little bit, and we have ATP hydrolysis. So this is the next slide. So we have this first binding step here. This induces ATP hydrolysis. We have phosphate release. The molecule sitting here, this means just that we have this binding process. Oops. And this distortion leads to an opening of the transporter. Water flows in, and due to the income of water, the molecule immediately turns around such that the hydrophobic part goes to the aqueous phase, and the hydrophilic blue part goes to the aqueous phase, and the hydrophobic part remains in the membrane phase. Then we have, this is ADP, ATP, these two states here. Then we have ATP rebinding. The molecule closes, and if there is something left, it's squeezed out into the lipid phase. So this is maybe surprising if you are used to the alternating excess model, but Camilo Perez and Locho also observed a similar or suggested a similar approach or a similar model for a glycolipid. And they were very puzzled at the time, and interestingly, they did this structure with two ATP analogs bound. So it was in principle this structure here which they presented in their X-ray structure. And so we looked at the kinetics of very many four transporters. So the P-glycoprotein, which was a gift, as I said already, from Michael Gottesman. We had CFTR, which works in a very similar way. Then SAF is an analog of P-glycoprotein, also with interleaved helices. And then we have BCRP. BCRP takes non-amphiphilic compounds, whereas PGP takes the amphiphilic compounds. And but the interaction is very similar, actually, in P-glycoprotein, CFTR, and SAF. We have amphiphilic molecules which probably bind in a similar manner. And yeah, that's so much for my presentation.
So thank you very much, Anna, for this very interesting presentation on the mechanism of action of P-glycoprotein. Actually, I think it's still compatible with the hydrophobic vacuum cleaner. It's more in the detail of what's happening.
I think the hydrophobic, the hydrophobic attraction is certainly there, but it's not to the protein, it's to the membrane. And once in the membrane, we have these weak electrostatic interactions. So the membrane is actually a hydrophobic vacuum cleaner, if you want. So I would call it an electric vacuum cleaner or electrostatic vacuum cleaner rather than a hydrophobic one. But of course, the hydrophobic effect, the first effect is still very, very important.
Okay, I've got one question in the interest of time from Ian. I'm reading up interesting work on it ties with some biochemical data we have on mutants of ABCD2 and ABCB1 with Rick Callahan, that there are superficial interactions between the drug and the protein surface via the lipid bilayer. Can I ask you to speculate as to why the EM and crystal structures pick up cavity-bound rather than surface-bound substrates? Affinity, lack of membrane in the structural studies? Maybe Ian will explain. Yeah, did you get the question?
The cavity bound, I mean, the point, are you thinking about structures?
Yes. I think this is Ian's question. Yeah.
I'm not, this is a difficult question. I mean, what I'm sure, you can never be sure. What we saw in our molecular dynamic simulation was, and we also analyzed a lot of deer data, and we don't find unambiguous behavior of the, is it closed, is it not closed? So it's open from point of view of deer.
Okay.
And the question, if I got it correctly, is does it go into the cavity of the small cavity of the transporter? Is that correct?
Well, it's rather whether the crystal structure pick up cavity rather than surface substance. But I think Michael Gottesman has a comment or a question.
Yeah,
Michael?
I do. Thanks a lot, Anna. That was great. I want, if I understand correctly, at the end of your second step, the substrate is sticking into the outer leaflet of the plasma membrane with a hydrophilic part coming out. And I'm going to suggest the third step. There's a lot of data, old data that no one's ever followed up, showing that the transport process is much more efficient in the presence of albumin in the extracellular space. So the third step may well be that you need an acceptor.
The extraction of the
extraction from the
I
membrane.
completely agree. We actually measured drug binding to albumin, and it's very much hydrophobicity or amphiphilicity is a big factor for albumin binding, and this is a very good point. I think Al-Shabi measured, you may remember, he spin labeled verapamil and measured, he wanted to measure how much gets out in the aqueous phase, and he had a quaternary ammonium ion on verapamil and thought it's so charged it must go into the aqueous phase. But it remained to 70% in the membrane and only the charge was sticking out. So I think it really needs something to extract it from the from the membrane. And there is another other point, this CYP, of course, also works in the membrane, makes it more hydrophilic, and then it will leave the membrane also. So there are many factors working on it.
I just wanted to defend the term hydrophobic vacuum cleaner, which I coined. The intent was that it was a vacuum cleaner that removed compounds from the hydrophobic bilayer. It itself was the vacuum cleaner.
Okay.
So I think that's consistent with your model.
Yeah. I just the point which, when I did this review, was kind of, I mean, I always propose this electrostatic interactions, but the point that when I looked at the numbers, that the epsilon, the dielectric constant, makes such a big effect in the affinity, and that you see this then afterwards in the rate, because as do you see this? Oops, no. Do you see the slide with the 3 transporters here? And so if the affinity here, it's a bigger molecule which has a lower weight, but the bigger molecules usually have a higher affinity. Now, if you think of a reduced affinity just because of the lower dielectric constant of the micellar environment, you have this lower affinity and you have a higher rate. And this is, I mean, the Suresh experiment and our early experiment in SAF. So this, I only calculate the numbers now, and this was, yeah, something which brought me to this model, actually.
I see Till Luckenbach has raised his hand.
Yeah, actually, I'd like also to ask you something. Thanks a lot for this talk, Anna. So when you say, actually you don't say it, but the others say vacuum hydrophobic vacuum cleaner, which but actually to be actively transporting or to be effective as a transporter, a compound cannot be fully lipophilic because it diffuses too fast through the membrane. So it needs to diffuse slowly through the membrane to be able to be actively transported. And I was just wondering, you know, so you need a compound that has hydrophilic and lipophilic properties in a way. And I was wondering, do you know how fast these compounds that you have as substrates or recognize as substrates, how fast they actually diffuse through a membrane?
Yes, if you see this slide here, you see the green, actually here on the y-axis is the passive influx, calculated passive influx. These stars here with the line is the transport activity. And on the x-axis, I have the cross-sectional area of the molecules. Now, passive diffusion is extremely dependent on the packing density of the membrane, on the size of the molecule. So it's an exponential dependence on the size of the molecule. And you have you see the molecules here, the small ones up here, they are extremely fast. So, oops, they are extremely fast and they go through. So green is for green light, you can walk, they just go. And the red ones have to stop because they are too big. Their passive flux is in the order or even lower than the active transport rate of the transporter. So the stars here are measured values for transport. And interestingly, the transport is much less packing density dependent. This may be strange for you when I talked about the micelle, but micelle is really something very different. The dielectric constant is very different in a micelle. And for the transporter, we have a factor, we have very little difference, because the penetration in the membrane is lower when you have a high packing density, but the affinity to the transporter is higher. So we have a kind of compensatory mechanism with respect to the packing density, which is not the case in the diffusion. There we have the just strong dependence, exponential dependence on size and packing density. So you can, in principle, roughly estimate, if you calculate passive diffusion. Actually, it's even easier. You can say all molecules which are larger than 70 to 80 square angstroms in diameter, they will not. Maybe I have one other slide. Yeah, this one. This is how I entered the field. We measured, this was together with Merck in Darmstadt. They wanted to know whether one can predict CNS compounds which penetrate into the brain or compounds which do not penetrate into the brain. They wanted to make antihistaminics which do not penetrate the brain. So we measured the air-water partitioning. So how well does it go to the air-water interface? And we measured the CMC of the compound, and we measured the cross-sectional area of the molecule also by surface activity measurements. And we found that the small compounds lower than 80 square angstroms, around 70 square angstroms, they penetrate the brain, and the larger compounds do not penetrate the brain. And I gave a talk at a meeting, and someone, I'm a membrane person from my start. And when I started out and I measured this, I calculated passive diffusion, and the person asked me, what about P-glycoprotein? Could it have an effect? And I was my most difficult question ever, because I had no idea about P-glycoprotein, and I was very much worried. I went home and immediately searched for P-glycoprotein. At that time, we had no Google, so it was a bit more work. And this is how I came to the P-glycoprotein to work on this interesting molecule.
Well,
So
I'm glad you found it, Anna. And thank you so much for a very interesting both talk and discussion.